Hello everyone, welcome you all. In this session, we shall look into mumps. It is a contagious disease caused by a paramyxovirus and it affects the salivary glands. Let me explain more about mumps virus, how it infects the cells, how it evolves, and how it can diagnose and treatment. Generally, viral disease or illness caused by viruses, which are very small infectious agents that can only multiply inside living cells. The mumps virus belongs to the rubella virus family of paramyxoviruses. It has helical nucleic capsid that contains single-stranded negative sense RNA genome and an RNA polymerase. The nucleic capsid is surrounded by a capsid and an envelope with two glycoproteins, a viral attachment protein known as hemocyanin neuraminidase and a fusion protein. The genome of mumps virus about 15,000 nucleotides long and contains 7 genes that encode 9 proteins such as N, P, V, W, M, F, H, N, L and SH. The 9 proteins binds to the RNA genome and forms the nucleic capsid. The P protein is the phosphoprotein that interacts with the N and L proteins. The V and W proteins are derived from P genes by RNA editing and have regulatory functions. The M protein is the matrix protein that links the nucleic capsid to the envelope. The F protein mediates the fusion of viral envelope with the cell membrane. The hemocyanin neuraminidase protein binds to the cell receptors and facilitates the entry of the virus. The L protein is the large protein that acts as the RNA polymerase. The SH protein is the small hydrophobic protein that may modulate the immune response. The mumps virus transmitted by direct contact, airborne droplets or vomitus contaminated with saliva or urine. It infects the upper respiratory tract and spreads to the salivary glands and other organs through the bloodstream or lymphatic system. The mumps virus replicates first attaching to the surface of cells with its hemocyanin neuraminidase protein, then fusing with its envelopes with the cell membrane with its F protein, releasing the nucleic capsid inside the cell. The RNA polymerase then makes mRNA from genome and replicates it. The viral proteins are synthesized and assembled into new virions that bud form the cell membrane. The pathogens of mumps virus cause inflammation and swelling of the salivary glands, especially the parotid glands. It can also affect the pancreas, testes, ovaries, brain and other organs, causing complications such as pancreatitis, orchitis, euphoritis, meningitis, encephalitis, deafness and sterility. The symptoms of mumps usually appear 16 to 18 days after exposure and include fever, headache, muscle aches, tiredness, loss of appetite and swelling of the cheeks or jaw. The diagnosis of mumps is based on clinical symptoms, epidemiological history and laboratory tests. The laboratory test includes detection of specific immunoglobulin M antibodies in serum or saliva, isolation of virus in cell culture or animal models, or identification of viral RNA by PCR techniques. Treatment. There is no specific antiviral treatment for mumps. The treatment is supportive and symptomatic such as rest, fluids, pain relievers, ice packs and isolation to prevent the transmission. Prevention. The best way to prevent the mumps is by vaccination with a live attenuated mumps virus vaccine. The vaccine is usually given as part of the MMR, that means measles, mumps, rubella, or MMRV, that means measles, mumps, rubella, varicella. It's known as combination vaccines. 2 doses of vaccines are recommended for children at 12 to 15 months and 4 to 6 years age. The vaccine is safe and effective and it can protect against mumps. Thank you for watching.